In this video, we will be looking at the molecular mechanisms through which the hepatitis C virus binds and enters cells. More than 58 million individuals currently have chronic HCV infection, according to the World Health Organization. HCV is one of the most common bloodborne pathogens, and its primary target is hepatocytes within the liver. HCV is a positive strand RNA virus, and its structure consists of an envelope, capsid, nucleic acid, and viral glycoproteins. A staggering 80 to 85% of patients with HCV cannot clear the infections and are burdened with chronic infection, which has many complications. Some of these include chronic liver disease, cirrhosis, and liver cancer. In fact, the leading cause of liver transplantation is chronic liver failure induced by HCV infection. For HCV to enter hepatocytes, it first has to recognize a cell by binding membrane receptors. We have two receptors on the cell membrane that are important for this step. These are the scavenger receptor class B type 1 and the CD81 receptor. HCV will interact with these receptors through glycoproteins E1 and E2. We are primarily interested in CD81 in this video, and this receptor is translocated along with the virus to tight junctions between the hepatocytes. Here, the virus engages with further interactions with occludin and claudin. The viral particle then enters the cell through clathrin-mediated endocytosis. Once in the cell, the endosome becomes acidified, which induces conformational changes in the viral glycoproteins. This allows the virus to interact with the endosomal membrane through receptors including CD81. This is followed with the fusion of the viral and endosomal membranes, allowing for the release of the viral RNA genome into the cytosol. The interaction of CD81 with the viral glycoprotein E2 plays an important role in the membrane fusion step, and this is what we will further explore now. CD81 is a member of the tetraspanin family and it is ubiquitously expressed in a variety of cell lines. It has 4 transmembrane helices, as well as a large globular extracellular loop made up of 5 helices. The extracellular loop is the segment that interacts with the viral glycoproteins. The molecular mechanisms that mediate cell entry and membrane fusion of HCV have previously been unclear. However, a recent study by Kumar and colleagues aimed to determine the specific molecular mechanisms responsible for this process. They showed that 2 steps are critical for the optimal fusion of the viral and endosome membranes. These include low pH within the endosome, and the interaction of E2 with a large extracellular loop of CD81. An extracrystal structure was solved for an asymmetric complex consisting of the large extracellular loop of CD81, the viral glycoprotein E2, and an antigen binding fragment of a non-neutralizing antibody used as a crystallization chaperone. Structural analysis has revealed 2 conformational changes in a region of glycoprotein E2 called the CD81 binding loop. In the absence of CD81, residues 384 to 421 of E2 are disordered, and a tyrosine residue at the tip of CD81 binding loop interacts with an isoleucine residue at position 422. This allows for the binding loop to pack tightly against itself in a retracted conformation. In the presence of CD81, residues 418 to 421 become ordered, wrapping around CD81, and the isoleucine residue at position 422 moves towards the core of E2, which disrupts its interaction with the tip of the binding loop. These conformational changes allow the CD81 binding loop to extend away from E2 and pack tightly against the large extracellular loop of CD81. Superimposing the previous structure onto the membrane-embedded full-length CD81 places the tip of the loop near the membrane. This suggests that the interaction of the virus with CD81 pulls the virus close to the membrane to support membrane fusion. The interaction of these residues is critical for the fusion of the viral and endosomal membranes. This is supported by structure-based mutants of isoleucine 422 on E2, as well as tyrosine 529 and tryptophan 531 located at the tip of the CD81 binding loop that block membrane fusion. The structural analysis is supported by liposomal flotation assays that show that low pH and CD81 interaction enhance the fusion of the viral and endosomal membranes. These insights support a model in which both acidification and receptor binding result in a conformational change in glycoprotein E2 that allow for optimal membrane fusion. Based on these new structural insights, we can investigate new druggable targets. There are currently no vaccines for HCV, and available drugs are only effective for specific HCV genotypes. For instance, Daclinza is only effective for 1 in 10 patients and only targets type 1 and type 3 HCV. Thus, prevention of HCV infection altogether using vaccines is an important area of research. The majority of currently studied neutralizing antibodies block the interaction of E2 with the large extracellular loop of CD81. However, as shown in the highlighted circles in the structural representation of E2, the regions targeted by these antibodies are not proximal to the CD81 binding loop. Based on the recent structural insights, we have a new potential target for antibody neutralization. At the tip of the CD81 binding loop on E2, 3 residues are conserved among the major HCV genotypes. These include tyrosine 529, tryptophan 531, and glycine 532. Given the finding that membrane fusion is blocked when these residues are mutated, we can investigate neutralizing antibodies that target the CD81 binding loop to block the interaction of E2 with the large extracellular loop of CD81. Thank you for watching this video.